Okay, so we're gonna get started. I just heard from Tony. Welcome everybody. This is our third or fourth broadcast for the Leguminaut Challenge, and the purpose of these broadcasts is really to reach out to teachers and parents who are trying to educate children or even college students, high school students from a distance. It's a real challenge. We're all learning as we go along and trying different things. And we've got a challenge here that I'll describe in a moment, but we're hoping that this might be an idea of something that you can incorporate with your students and engage in meaningful ways and keep the authentic science and math and learning going through the rest of this COVID lockdown. So, what are leguminauts? So legumes are a special group of plants. I've got one here. This is a cowpea plant that's about 5 weeks old. Legumes have the ability to form a symbiotic relationship with bacteria in the soil called rhizobium. And that's really critical because that is the only way really that we get nitrates and nitrites into the plants. The nitrogen that we're all swimming through constantly in our atmosphere is inert. It can't react, so we can't form proteins with that. These bacteria, rhizobium, are able to convert that nitrogen into ammonia and then into the nitrates and nitrites that plants need. And they do this through a special relationship with legumes. They form a symbiosis, a mutual agreement, where the rhizobium give the plant the nitrogen compounds that it needs, and the plant gives the rhizobium the carbon compounds, the sugars that it needs. So it's a good deal for both organisms. And while the plant's alive, it pretty much uses all the nitrogen that the rhizobium can produce for them. But then we've got that organic matter that once it breaks down in the soil, that those nitrogen compounds are released back into the soil. And farmers have been using this for a very long time. We call it crop rotation. And you might have a season of a couple different legumes and then a nitrogen intensive crop. So that's what we're doing here on Earth and have been doing for a long time. We tend to use chemical fertilizers or anhydrous ammonia in fields for high production. And we're thinking about, though, cropping off of Earth, being able to sustain space explorers on a long-term mission to another planet, perhaps, or to establish a settlement on the moon or Mars. You're not going to be able to transport all the food that you need. And it gets pretty boring eating prepared food all the time. Taste becomes a real issue in microgravity because of the fluid shifts, and it's kind of like trying to enjoy a tasty meal with a head cold. So we need things that are flavorful. We need good, variety of textures. Humans, we kind of stop eating when the food's not interesting. So we've got to keep all that stimulated. So we can't send the pre-made food or all the ingredients up. It's too expensive. The same goes for sending the fertilizers. So we need a biological solution, and we can grow plants in space. We've been doing that for a while. And what we don't know is, can we get this interaction between the rhizobia and the plants to work in microgravity? And then providing our resources to grow more nitrogen intense plants like peppers or grains. So that's kind of the science behind this. And this we flew through our seventh mission. Was looking at cowpeas and the rhizobium to see if they could nodulate on orbit on the International Space Station. That experiment concluded yesterday, and we'll be returning to Earth in a week or two. And then we'll see what happened with that post-flight. At the same time, I was growing plants that had been inoculated here. This seedling is one that started out in the test tube inside the exolab, and as it got too big for the test tube, I transplanted it into a simple growth system here that I'll explain later in the broadcast today, so that you and your students can make these on your own. But this plant's been doing really well. It has some flowers that this one is just looks like it started to produce the ovary started to transition to a pod, but it's got several pods on there, so it's doing well. What we want to see is, are there any rhizobial nodules on the roots? So we're going to go through a dissection. We'll uproot this, extract some root material. I'll show you how to make a system like this for growing the plants, and then we'll transplant this one. Hopefully it'll survive so we can keep producing its seeds, and then we'll focus on the roots. So I've got, let's put the camera that has the plant, just the plant here. This is kind of an overhead view, you know, like a cooking show. So I'm just gonna set the plant aside for a minute. I've got just
I like
real
your
simple.
setup.
Say again?
I said I like your setup.
Thank you. So these are just really simple materials. And that's the point. Anything that we're doing through this series of broadcasts, we want it to be easy for students to replicate at home. Welcome, Renee.
Hey.
So it's basic materials. This is a seedling that we'll maybe replant later. So I'll set that one aside. So I've got just a repurposed containers. So I've sterilized by washing them in some just water in the microwave for a couple of minutes, and so that they're transmitted.
What's wrong with my computer?
Got in one bucket as well, so I can just dump everything in there that I don't.
Let me get rid of Facebook. Could be that. Uh-oh. Hello?
Oh, we lost Michael. Hold on.
Okay. I thought it was my computer because things are muting a little bit.
That's okay. Hey, guys.
There you are.
Good afternoon, everyone. Oh, Michael's back.
There he is.
It's been a fun adventure in hardware at home. Welcome to the second episode today. This afternoon,
I have an unstable internet connection. So
Michael, welcome back. I'm on a Chromebook without a power supply, but that's a different story. You've got the helm, Michael.
Oh my God, these dogs.
Looks like he's frozen. We'll wait until he comes in. Renee, good to see you.
I see you too.
How's your world?
It's going okay.
Good deal. Good deal. You know what? I love it. Every single teleconference so far has included doggies. Right now, it's 5 dogs. I've had 3 dogs and 0 cats. There are no kitties.
Oh, my cats are downstairs. They're shy. They don't want to.
Oh, there you go. Yeah. Yeah. So the computers, work from home. It's fun setup. So I'm downstairs in my garage for the first 2 events we've done so far.
How do you read?
Oh, he's back. Hi, Michael.
Oh, you're freezing again, Michael.
Oh my goodness. I think it just might be one of those days. Yeah. So I guess I was losing my cool a little bit for a moment.
Okay, so I've missed here. Do you read me?
Yep.
Oh,
We can
Okay.
hear you now.
we hear you.
All right. So we'll try this again. I shut down my other computer. Let's see if we do better. And I'm just going to broadcast from the iPad. That's my overhead view here.
That's a great view, Michael. Thank you.
Yeah, thanks. So to recap, I've got materials here that would be easily found at home. And that's kind of the point of all the activities that we're going to be sharing through these broadcasts. We want this to be as simple as possible and things that students can easily do without a lot of supervision. And so I'm using recycled or reclaimed materials, repurposed materials. So I've got a sterilized container that has clean water in it. And I've got another one that's just my waste container that I can toss everything in as I go along. I have my seedling here that it's going to be our subject of investigation today. Pair of scissors. These are barbecue skewers, tweezers, and I do have a spatula here. I may or may not use it, but you could use like a table knife perhaps to substitute for that. But this is just down here on my desk. I also have little containers that cat food comes in. So these have all everything's been cleaned out, and these just make handy pieces of equipment to work with. So let's start by finding out if our cowpea seedling has any nodulation. I've already washed my hands. All of our students should be well versed in how to do that properly by now. And I'm just putting on gloves just to reduce the chance of contaminating my plant, picking up any mold spores or anything like that from the environment. So I'm going to lay the plant on its side. So I want to minimize the damage to it and gently pull it free from the container. Now this one I did use soil. I prefer vermiculite because it's a lot easier to remove it. Also, I'm thinking that this ridged bottle may be holding on, or I've got roots bound in my wick. So I'm just going to check that out. Yep, that's exactly it. So I don't know if you can see there, the roots are clear down here, and they were in my wick. So now I think we can get it free. Yeah, there we go. I'm going to try to get the whole root system out. There we are. Okay, so we don't need all this soil anymore. So I'm going to throw all that, and I've got another one of these cups here. So I'm just going to scoop all that in here, get some of this out of the way. All right, so what we're looking for are little nodules that are on the roots. So I'm just going to gently tease away soil here. I want to keep as much of my root system intact because I am going to try to replant this one. Okay. Yeah, I think I see some here. Yeah, there we go. So I'm just using that barbecue skewer as a probe. If I were back at school, I might have dissecting probes handy, but your students probably aren't going to have those. So I'm trying to model everything today with materials that you're likely to have at home. Okay. I'm going to try washing this. So see if I can see things a little bit better. So I've got my water, and now I've got mud. Okay. So we've got most of the soil off of these roots now. And I'm just protecting my desktop with a blue chucks. Like if you got pets, you might have these around. for house training. Okay, there. Now this one doesn't have a lot of nodules, but I do see some. So what I'm looking for are these small spherical objects that are clearly attached to the roots. And I don't know if you can see them in this view right now, but what I'm going to do is I'm actually going to clip some of these free, and then we'll be able to look at them more closely. But you're going to see that they're attached directly to the roots. So I'm just going to clip that piece of root. And bring that up to the camera, so hopefully you can see.
Maybe if you put your gloved hand behind it, it's good. Oh, there you go. Great. Thank you, Michael.
Okay, so there you go. So you can see the nodules there. So this is a really cool interaction. The rhizobia that I talked about that's in the soil, it is initially a pathogen. It's an infectious agent, and it literally infects the seedling's roots, and there's a response like we all get from an inflammation response, and the seedling actually wraps around the rhizobia and creates those nodules. So it's on the really fine root hairs in the beginning of the plant, and that's where the nitrogen fixation occurs. So it's got a direct line to the plant system. So I'm going to take my cat food container, put a little bit of water in there to help rinse that out so we get really clean. And then we'll take a closer look at that in a few minutes, because this plant is not going to be happy with its roots exposed to the air. Take a closer. So I'm going to make my planting system. And this is going to allow me to keep my plant well watered and minimize the maintenance. And again, it's just recycled materials. So I've got an empty bottle, washed it out. I'm leaving the cap on because that's going to be where my wick goes. I'm just going to take the point of my scissors. Also, if you have a lighter and a nail, that'll just zip right through. I made a small hole, and that's what I'm going to put my wick through, which is just some cotton string. And we'll get back to that in a few minutes. But I want to think about, okay, where am I going to cut this? Because this bottom part is going to be my water reservoir and the base for the whole system. The top part is going to be my pot. So I'm going to cut it right about here where this transition in texture is with the utility knife. And if you can see that, there we go. So I'm going to just get it started. And then once you get the blade of the knife through, this plastic is really easy to cut if your knife is good and sharp. You might want to have your students use a scissor. It's a little safer. They can just work their way through it. That's fine. I just find it a little bit faster for my purposes with the knife. All right, so there's my water container. And what I will end up doing is I'm going to push the top of this bottle down in here so it goes all the way down. There we go. But I need to get my wick in there first. So I'm going to pull that back out. The wick is just going to be a piece of cotton string. So I went to the kitchen and got my poultry trussing string, my kitchen twine. I'm going to put my twine. Sorry, the bifocals are messing with me. My twine in the hole. And I might need to make my hole just a little bit larger. Okay, so I'm going to pull that up through there. I want plenty of it hanging below the cap, and a fair amount of it coming up through, so that that ends up in my potting media. So I'll just thread that back in. Okay, I'm going to hold the string with my hand so I don't lose it, and I'm going to put my wick down back in and push it all the way back down. So I'm just getting my fist in there and pushing it straight down. Okay, nothing, it's not real elegant, but it works. Now, the potting media, as I said, I like vermiculite because it does not cling to the roots. And if I want to take this back out and harvest some more nodules, it makes it a whole lot easier. So I'm just going to pour a little bit of my vermiculite in here. Okay. I'm going to make use of my muddy soil. It came from the roots. And I want to make sure I've got enough here for these roots. Okay. So now we're going to place that plant back in. Get the wick with the roots and fill it up. Okay. And just pack it a little bit. My first node where the cotyledons were, it's starting to get some leaves there, so I don't want to bury that. I'm just going to come right up to it. And now I'm going to pour some more water in there. And I want to add water carefully and slowly until I see water that's starting to fill up down in the reservoir here. Trying to get an angle so you can see. The nice thing about this is it helps prevent overwatering too. A lot of legumes do not like to have their roots totally wet all the time. A little bit of settling. And you can see I'm starting to get some water down in here. I'm going to add just a little bit more because then it helps. I don't have to mind it so much. The string will wick up the water. Okay. So hopefully it's not too worse for the wear. And I'll put it over here under my lights. I've got some fluorescent, high intensity fluorescent lights that I just keep on 24/7 for those more mature plants. But you can put it by the window or whatever made sense. So any questions about that system? Okay. So Ted, or Tony, if you're on, what I would like to do is get the nodules excavated here so that I can get a decent image broadcast. And if you can tell people a little bit more about the program, show them the platform, I think that'd be a good time to do that now.
Okay. One moment, please. Ted, you have something else to add?
I do. I want to start with an apology.
Yeah,
thank you, Tony, and thank you,
getting
Michael.
on right now. So go ahead and fill in the blanks.
Hopefully everyone can hear me all right. I want to start with an apology, frazzled nerves, I guess, here, working to make sure that we all kind of come together and share some amazing experiences. Michael's walking through that kind of dissection of the plant, which is fantastic as we think about something that's grown fully. And really that symbiotic relationship between the rhizobia bacteria and the legume is really what we're hoping to replicate in microgravity. And what Tony's going to be setting up for us is really a pretty amazing experience. We understand that there are districts out there and teachers that might have difficulty getting all their students online. So what type of experiences can we create? Can we share with you? Can we collaborate with you on to provide a way to engage your students without a lot of digital, right? How can we do that analog? And I should, I guess, point out that Magnitude, we do send things to the International Space Station, and we provide a relatively live telemetry of various sensors of what's happening in the environment for the plant, including imaging, and that's sent down to classrooms really around the world, Africa and in Europe, here in North America. And so the challenge we're facing here is with the everyone kind of having to be at home and our school shutting down is how can we reach out to students? If students are you watching, thanks for joining in. And for you teachers and parents, I think we can find some practical ways to get these things set up. So there's the platform. Tony's going to walk through it, but I guess I want to complete with my apology. As this video started off, I kind of was losing my cool a little bit, trying to make everything work here at the office, and 3 different computers aren't working for me. So I kind of, maybe I need to get a little bit of rest myself. But I'm going to leave the helm back over to Tony. He's going to walk through the biological design challenge we've set up with Exolab. And with you, we have a task. We've got to try to find a plant to send up. And actually, it's now been moved to October as we got notified by the National Lab. Our SpaceX 21 mission, originally scheduled for August, is now October, which, oh, there's always silver lining, right? In a cloud means we have a little bit more time to experiment and think about reasons why we want to fly a particular legume. Tony, why don't you walk us through the different choices we might have? Thanks.
So I'm going to start from the very top. For someone who are new, wanted to join us, right now is a very good time to do that. We have set it up in a way that you can set up a free account. And within it, there are some free lessons that is related to this challenge on the platform. So all you have to do is really go to Magnitude.io. And right on the home page, you can just click on the purple button right here. And it will take you to the landing page. So we just recently made some changes to the landing page. The first thing I wanted to point out is that the launch for August has been pushed to October now. I believe is the end of October. So like what Tess said, we have a little bit more time to prepare our experiment and bring some additional support from Japan or Wisconsin. Now, one other thing I want to point out is that now, if you have missed some of our earlier conferences or video conference, all you have to do is come to this page and you can click on these 2 buttons. Now, in order for you to sign up to our platform, it's quite easy. All you have to do is click on the free sign up. It will take you to a sign up page, and you can, whether your school email address is created by using Google or Microsoft, just select the button and use the email address that you were assigned with your school, and click next. I am going to select my own district. Now, if you don't have a district in our system, all you have to do is click create new school or new district, and it would prompt you to the right area. So you click on that. Okay, I'm getting that because I have used this email once before. So it is asking me to just go ahead and log in. Now, for someone who is using your email for the first time, you will not get that that message. So what you're seeing here is the dashboard. Now it has some additional information across the very top right here, and it will really guide you through how to get started. For example, the first thing you want to do is create a classroom. It's quite easy to do, and just a couple of fields, and you are done with creating your own classroom. Now, once you have the class, okay, I'm just go ahead and create a class. Once you have a class created, you are given the class code and a special URL for your student to create their own account for your classroom. So it's quite easy to do. If you want to upload a CSV, you can do that as well. So over here, once you have the classroom set up, now your student, when they log on to their own account, there's nothing for them to do until you assign them a lesson. So by clicking on this button, you are able to access all the free lessons that we have set up for this one challenge. You can simply click on the lessons to see the entire lessons broken down into different sections. Now, each section is guided to in the student account, so they have to finish one section at a time. Now, once they are working on the lessons, going through the process of the lessons that you have assigned to them, you will see all the progress, real-time progress showing up right underneath here. I will show you that in a moment. Now, yes, you are able to see some of the tiles or the different lessons that we have set up for different experiments. And you can and now my lessons is very cool. I just wanted to show you that very quickly. For example, this one lesson, you notice, okay, my grade level is maybe a little bit too low for this, for all the wordings that we have set up here. What you can do is that you can clone this lesson, call it my first clone lesson. Once you have this clone, you can you go to my lessons, and you can modify this lessons entirely to how you want to teach it, or you might want to make changes to all the characters, or maybe you want to translate it into Japanese or Spanish. So you can do all that entirely yourself. Now, once you have done that, all you have to do is assign that to your class, and your entire class will start using the tailored lessons that you have just made. Any questions, Gil, about the platform?
No, it looks pretty great. I'm just I'm thinking about the translation part with the so if I have the initial account open for the class, would I be able to put in other teachers so they could work on translation work on it?
We will have to do that with you. That's not a problem.
Okay, cool.
Yeah,
looks fantastic. Yeah.
Quick question.
Sure.
Yes. So if we've already got Exolab, because we've been on Exolab, doing what we were doing before, Does that mean we already have access to this and we don't have to sign up for anything? Because it looks
That's
that way when I
correct.
was just going
That-
online with it.
That is correct. All the free lessons are available for all the existing teachers as well.
All right, great. So we can just dive in when we're ready. All right.
Exactly.
Okay.
Yeah, please use them and give us feedback. And we would love to tailor um, whatever you think is best. Yeah. Oh, let me show you one other thing. You know how you monitor in real time. So I'm going to log on our demo site to show you a teacher who has assigned a number of different lessons to their classes. Now, in this view, this teacher has a live view of how each students are progressing through all the different lessons that they have that they are assigned to. And if they have, if the lessons has a number of quizzes embedded, you will see the answers to their quizzes over here, if they have gotten it right or wrong. So you can make an adjustment to it. So if you have a essay test within the lessons, you can view their answers over here. So really very easy to do. Now, if they are taking a lot of notes, and I do encourage everyone to really use the notes section. For example, right here. Okay, I have to switch back to the other one. Okay. Are you still seeing my screen? Yes. Okay.
Yeah.
Let me switch back to the other one. So when you are in the lesson, all the students are able to access this notes section. This notes section is a really good tool for them to take notes. You know, for example, they are making observations on the plant, and they want to take notes each day. This is where you want them to type in their notes in their own words. Now, for example, if you have given them a assignment to do some research on a particular word or plant, they can put their entire research in the notes. If they have screenshots that they wanted to store, all they have to do is drag it into this white box right here, and our platform will store everything for them. Now, at the end of the semester, this can become a very good tool. For example, if you have a science festival or science fair or open house, all the parents are coming over, to the classroom to look at all the experiment that you have done throughout the entire year. Now, the student can just go to the notes sections and download all the notes that they have collected throughout this entire experiment. Just by clicking a button, all the notes will become a PDF for them. And they can print it, enlarge it. So it is an excellent tool for them to, really take good notes. I think that's all I have for now. Michael, do you want to take over?
Sure. So let's see if we can share this content. There we go. So those are our nodules. The grid is a 1/4 inch grid. So those are approximately 2 millimeters in diameter. You can see they're completely fused to the root there. And that's because that lump was a little root hair that's just curled up and then created this cyst. Now, what we want to see is if these are active or not. The really cool thing is that when they're active, they produce a hemoglobin-like substance that reacts to oxygen. So it's going to turn bright red if they're active when I open it up. So bear with me while I slide this out from underneath the microscope. And I'm just going to cut in half. It's just an X-Acto knife. Okay, and I'm going to place it. Where'd it go? There it is. Yes, it's active. Okay, let's see if I can get this in here. And where is it? There it is.
What happened?
I'm seeing some color there. That looks good.
There we go. So you can see the center of this is active. It's red. And can anybody remind me what the name of that is? That Gil, do you remember? It's legumoglobin or something like that.
Sounds about right.
As in legume?
Yeah, I don't know why, but that's just one of those terms that just I cannot get to stick in my brain. I've looked it up so many times.
Yeah. What we'll do is we'll include a link.
Yeah. So you can see the tissue, the lighter tissue around it. That was the root hair, and the rhizobial colony that's completely undergone a physiological change. Both have actually. So we've got this active nodule, and that is where all the nitrogen that we all depend upon comes from. Every living thing is made of protein. All proteins are nitrogen compounds. No life can exist without this symbiotic relationship, because the only other way we can get nitrogen fixed in our atmosphere is through a lightning strike. And you know, there are some places around the world where lightning is very common and you get lots of strikes, but that's not true across the planet. So we have to have these soil bacteria, the mycorrhizae. That top few inches of the soil is not an inert thing. It is a living, breathing organism almost, or a superorganism. This is that, and that's the difference between regolith and soil. This is living stuff. The regolith, you can think of that as just sand. It's minerals, salts, things like that. Nothing organic in it for the most part. So one of the things that we're thinking about with this investigation is, can we actually produce a living soil from regolith? That's kind of where our long goal with this experiment on the ExoLab, and legumes are the plants that do that.
Yeah, legoglobin is a shorter definition.
Legoglobin. There.
I put a link in Wikipedia there. We'll put it on our YouTube and Facebook. And I'm on a temporary Chromebook right now, so I realized I got bumped off and back again. I'm going to go and drop my video to save the battery. Michael, this is fantastic.
Thank you. And you could zoom in further. You can actually extract the rhizobium from these nodules and grow it on media, on a petri dish, and get pure colonies of it. That if you have older students who are doing real microbiology, these are gram-negative rods. You could go through the whole identification process and actually should be able to identify the biovar. So this is Rhizobium leguminosarum. I think this is the biovar. No, I'm not going to say, because I think I've just drawn a blank and I don't want to be Yeah, I'll have to look it up. But it is the one that is particularly well suited for cowpeas, most of the vigna, some of the clover, and that's one of the interesting things is every legume species has its preferred rhizobial species. So you can inoculate soybeans, let's say, with the rhizobia that you harvested from a nodule of, let's say, from a pea plant. And there's no guarantees that it's actually going to nodulate because that might not be the right species. So as we think about moving into using these green fertilizers on Earth and using them on the station or on a habitat on the moon or Mars, we are going to have more than one kind of legume to make sure we get all the varieties of rhizobium that we need in order to produce all the nitrogen, and also just for those plants to be healthy. Because if a legume isn't able to nodulate, it can get nitrogen from the soil, but it's not as efficient, and they're generally not going to produce as much fruit. Questions, comments? Yeah, Gil, go for it.
Michael, by the way, this is just great. Sorry if I sound kind of weird. It's just turned 7 in the morning over here. Have you guys already done extensions for legume preparation with students?
In terms of preparation for what? I guess I'm not following your question.
So a lot of the food on station and a lot of the literature right now, they're talking about crops that are pretty much they're grown and ready to be harvested and eaten. And with legumes, there's just preparation. And I'm just hearing you talk, I'm thinking this could also be a really cool opportunity for students to kind of brainstorm if we had a bunch of legumes in space in a limited environment, how they would go about making them viable as food sources.
Yeah. Now, presumably on a settlement on the moon or Mars, we're going to have more legitimate kitchen facilities than we do on station. But the astronauts are pretty creative. They take those components of the different foods and come up with some whole new combinations and things that like substitutes that they don't have the full ingredients to make something in particular, but then they combine a variety of things and come up with something that's kind of like. I think they wanted burritos or tacos or something, and they took a variety of different parts from other foods and put those together because they didn't really have all the ingredients for the taco. Yeah. So yeah, I think one of the cool things is some of these plants, like the shoots of the early beans, those are edible by themselves, and you can eat them raw, or you could steam them. You maybe will have a facility of a microwave, but you can definitely make warm water, so you could warm it up at that point. And honestly, other than lycopenes that are in tomatoes and spinach, most nutrients are more concentrated and of better quality with the raw foods. So all those that are eating spinach salad thinking you're doing a good job on your lycopene, it hasn't been. You gotta cook it before that actually works. But I don't believe legumes fall under that. And then of course we have the beans themselves. A lot of people eat the soybeans. You just blanch them in some hot water and eat them raw, shell them out. All of those things are possible. Some of the pea, like really new peas or clover or even alfalfa, the shoots of those, forget growing the full plant, just the seedlings, they're great microgreens, and those are packed with good nutrients and fiber and taste. So there's lots of different ways in terms of what's going on. What I've done with my students is we always, whenever we're doing an experimental plant, we always grow those same plants alongside them on a larger scale so that we can do taste tests and come up with some creative ways. So when we've done different greens, they've tasted them individually and done taste test analyses, and then they've mixed them together to make a salad and, you know, do a simple lemon and water dressing if they want something, but most of them find just plain is fine. And like we grew wasabi, and that just added a great little bit of spice kick, and so the students all were positive. Wasabi is something we've got to grow in these microgravity environments, because the astronauts all talk about losing a sense of taste, so they like spicy things, and wasabi would totally fit that bill.
And I think that was Flight 5, Michael. We flew wasabi along with amaranth, pak choy, and another, what was it, Mizuna, I think.
Yes. No, 4 was Mizuna. 5 was pak choy, wasabi, amaranth, and wasabi. Did I just get 3? I'm leaving something. Well, purslane,
yeah.
Purslane.
Which is notoriously difficult to get to germinate. So these teeny tiny little seeds, and we've had a hard time getting them to germinate. So Tony, I tried that was with this little microscope. So it was pretty decent resolution on that.
That's what you were using.
That's what I was using. I thought I would
the Lego
my regular microscope and put the camera up there, and I got it. But then I also realized, well, shoot, I've got this little toy. I can play with that.
There you go. That's fantastic.
It worked out well.
It is. Now, I guess if we're not seeing that reaction, but the nodule is developing, what does it look like on the inside?
If the nodules aren't active, then they don't get that pink color.
The nitrates,
so that's the first thing. And then if we don't get nodulation at all, then whatever, if we know we put rhizobia in there, we've got 2 possibilities. It wasn't a viable strain, meaning it wasn't vigorous enough to actually survive and infect the roots, or we had the wrong strain. So then you'd want to go back and test for those possibilities. Maybe try a different strain, maybe try a new culture. All of these things you do have to keep them fresh. They have a limited shelf life. So that also will be something that we have to think about on space. So probably the astronauts would have to harvest some of the nodules and actually culture them to keep a good supply of these organisms.
Oh my goodness, Michael. Can you imagine actually that it'd be an experiment in itself of the mature nodules developed here on Earth sent up for replication in space to basically grow a colony of rhizobia, right?
Yeah.
Microgravity rhizobia. How might they be different? Now, we've been focused on the legume, right? Oh, we're off the deep end. We should probably weave back to our challenge because we really want to make it easy for the teachers and for the students out there in light of everyone kind of being at home or in a garage, as the case may be. So what is the challenge about? We want to find a candidate to fly now in October, so we've got some more time. What's going to make a good candidate? What's going to be a bad candidate? What are some criteria? Gil, Scott, you guys are on. Renee, you're on with us. Well, as educators, what are some questions you think your students might be coming up with?
Yeah, I think something that we've been driving home a lot in our astrobotany program are what are the significant factors that make a crop viable and useful in space. And one of the things that we've focused a lot on are crops that are nutrient dense. The value of nutrient dense crops, that's why a lot of our studies have focused around microgreens, because of the high nutrient content in a lot of these little plants. And I think within this challenge, something that I would encourage everyone to really look into, it's like, if you had a deficiency of something, of a nutrient in your body, whether it's a vitamin, whatever, what would be the most efficient way to get it through your diet, and which one of these crops could facilitate with that? That's a challenge that is not only useful and necessary in space, but it's necessary here on Earth. And ultimately, everything that we develop for our space programs, the food production procedures and protocols can be adapted to places here on Earth where there's food scarcity, where people are not getting their proper nutrition, um, and into more sustainable agriculture as a whole. So, two things that always come to mind in our program is um, nutrient dense and how to get those awesome nutrients, and the application of it to impact humanity here on Earth as well.
You know, that's a great a perspective. The International Space Station, the U.S. National Laboratory, which we're very grateful for, we really wouldn't be able to get our experiments up there without the National Lab. It's their mission is to make life better here on Earth. While we dream of the stars and getting settlements on the surface of the moon and Mars, I mean, how cool is that, right? But you know, let's talk about nutrition and health and well-being for the nearly 8 billion that are here on the planet. So that's an interesting perspective. How much water does it take? You know, is it can it handle brackish water or kind of saline or water that has a lot of salt in it, or other salts? So really, really fascinating. Gil, you're joining us from Osaka, Japan, which is pretty awesome. So thank you. Tell us a little bit about your school, and it's pretty unique. It's a middle school and high school. Is that correct?
Yeah.
Yeah. So share it with us a little bit.
Officially, the school's doors have been open now for a full year. We're about to start our second cohort, but it's been in development for years. And I was part of the development team, and part of I am the head of science at the moment. Basically, the school is it's the first public school set up by the Osaka Board of Education in Japan, that is owned by the government, operated privately. So the Osaka International YMCA Education Department runs it. And what the interesting thing about our program is that it's a junior high school and a high school, and the English, the science classes, and the English language classes are all offered in English. And not only that, the IBDP program is also offered and available publicly. So it's a program that allows students that that aren't able to go to these really expensive international schools. They get all the facilities and all the program access that you would at an international school at a local public level. What's cool as well about the science part, and I have to cue that in because that's the program that I developed. It's primarily taught in English, but we have 4 different labs that are fully stocked, for classes and for research. We just got a PCR machine, and we're connecting with universities all throughout Japan to get students not only meeting their national requirements, but also conducting significant science that go beyond an assessment and actually feed into active data pools that are influencing research and industry.
Well, that's fantastic. And you actually are connected in the Gilroy lab in some way, right? Dr. Richard Barker has been quite entertaining in his deep dives and explorations of the sciences and thinking about what's possible.
Yeah. So our school, we have the only astrobotany research program outside of a university in Japan, and that's the program that I coordinate and direct here. So it's like the Suito Kokusai Astrobotany Research and Education Program, and in the development phase of that, we needed more expert help and guidance. And I reached out to Dr. Gilroy, met with him and Dr. Barker, and they were so generous and so open that we just became partners. And the program that we've developed in Japan basically runs the experiments and the protocols and stuff that they've already developed. So we continue on what they've started, while at the same time we provide them with valuable data sets and educational models that they can then give out to other people interested in running this kind of program in their schools. Currently, right now, Dr. Barker and my team, were developing an international astrobotany research initiative which uses some of the tools that he's developed, like Mango, which he was talking about yesterday on the ISS. I'm sorry, not astrobotany, astrobiology, which includes astrobotany in there. But yeah, Mango, and the growth of plants in these microgravity stressor chambers that they've developed, that you can either print where you're at with a 3D printer or order through Cozy, which is a company they kind of developed as well. But yeah, it's been super fun working with both Richard and Dr. Gilroy. They're both brilliant. They're both very, very interesting characters. Like
I'm starting to figure that out.
Yeah.
Dr. Gilroy is actually called the Wizard, I guess, right?
The Wizard, man. They call him. I mean, the first time I met him in person, I was just like taken back. We were like right outside of Kennedy Space Center, and there's this brilliant guy who's like the head of so much in terms of astrobotany and what's happening in space right now with plants. And I'm nervous about to meet him and Dr. Swanson, and here he comes up wearing like a hula shirt, and he's got this long white hair, and he's like, hello. It's just super like energetic and open and passionate.
Cool.
They're just warm, caring characters that love what they're doing, and they love getting this getting as many people as possible involved.
There you go.
So
you
guys don't know yet, check out Gilroy Lab and what they're doing. They're fantastic, and they'll talk your ear off if you.
That's right. Well, I'll tell you, Dr. Barker is quite persuasive. We're going to already be looking at some interactions together. I think cool people really love plants and animals. And Renee, you've got like a completely different experience working with homeschoolers out there in the Northeast. So tell us a little bit about what that's all about. Here, you're kind of all of our teachers that have that classroom that they go to, and the kids sit down and they go to the next class, or they get them all day long. That's kind of a routine, and now that's kind of been disrupted. And here you are with your center that's set up, and you kind of work already in this kind of environment. So, and you picked up ExoLab, you're starting to work and seeing how it interacts. What are some of your thoughts about this?
So, we moved with homeschool first to kind of, for me, a learning experience, and the homeschool's been engaged. And, obviously when we're thinking about these plants from the audiences that I work with, it's more simply with, well, why are we picking this? What does this mean? Learning about the plants and just kind of going through the process to kind of help them understand. And it was great hearing from the families how serious they were taking, and still are, some of the challenges they run into with preparing the seeds. And I'm hoping that it does get challenging, because we're trying to look at, okay, well, what can we do as a museum? Because ultimately, even if and when things improve, it's probably going to be months before they'll even allow us to open. They're saying probably August, maybe, that we'll be able to reopen. So I'm trying to think about, well, what can we do that's meaningful? There's a lot of people right now doing quick videos of trying to show them being out there. Fortunately, I can go back to the museum. It's one person. We only can be there for like 4 hours with gloves and sanitizer. And, even at that level, because we're at the point things are increasing a lot. I'm sure, Michael, I know you're in New York, things are kind of in the same place. So I'm trying to see how can I get involved with the digital, bring this to schools, because now they do have to do this online.
Right.
And I know next week for us here is kind of the first go in Rhode Island with the districts. So I'm trying to get actually from the state of Rhode Island level with the education commissioner, actually going to her to see if I can engage or, work with one district or a couple of classrooms, because everything right now is actually being driven by kind of the district level. So, which I think is more challenging for teachers, because, especially when you think about some of the elementary, they're not using some of this technology. Right. So I know that Monday's the start of this distance learning for, using Google Hangouts. And right now people are excited, but I'm just curious after next week happens, that they're going to be hoping or looking for some of the things like what Michael did today, and just kind of showing that. And, I really didn't have a chance to look at the curriculum for the challenge, just because I've been on, emergency planning and documents for if I can't go into work, who can go in the museum, where's the, those type of contingency plans and planning, and all of my staff's working remotely. So that's been kind of dedicating my time. I've actually been working more than ordinary on things that I wouldn't have to. So it hasn't been the fun stuff, but I think even just bringing some of these lessons to the schools, and then even if they watch online or actively engaged, I know for some families, they don't even have some of the things that even Michael showed. So, just observations and science process skills, I think will be helpful. So I'm trying to find a way that I can bring this to them. And again, I'm most of my audience is going to be elementary. And with the homeschool, we have some high schoolers that do get this a bit more and asking great in-depth questions. And I still want to do kind of a Zoom call and some things like that. But, that's kind of where I am. And I kind of straddle both worlds because I'm also in a public school district, managing a planetarium that, the whole second half of the year, that curriculum that I do with them isn't happening. So trying to figure out with Middletown how I can bring some of that to them virtually is what I'm working on as well. So.
Wow. Well, we've got some ideas, but hearing from you in that role, I think is really helping us at Magnitude understanding where the need is and what we can provide. And we're trying to look for a lightweight, engaging solution and almost taking testing and standards and throwing them out the window, saying, let's be curious. I think at the federal level, it looks like they are willing to take state testing and federal testing and kind of putting it off to the side because of what's happened. Scott, you're in a public school in Southern California, and it's a smaller school district outside of the Los Angeles immediate region there. What are some of your thoughts, and how does it affecting your community and your parents and you as a teacher?
Well, obviously, we're suddenly thrown in, same as the other two, we're thrown into sort of this almost crisis mode here. So the first day after the Friday, we come back in and we just throw together some kind of lesson plans so that the parents and the children who aren't online have at least something. Now, we're starting to think more long term. We're setting up the communication. We set up the communication basically, but it looks like it's going to be a lot longer because originally we just set it up for 3 weeks. Now it looks like it might go till the end of the year. So we're also moving from sort of an enrichment review sort of mode, which is where we were before. Well, we'll come back after spring break and we'll in there. I think we're going to need to go into learning how to transmit new material when we're not physically present, which is always really a challenge, as you can imagine.
One thing that I've been thinking about here and the team is the analog, right? Not every school district has one-to-one. In fact, most don't. One-to-one being, a computer for every student. Some students, even in a one-to-one district, don't have internet access. And of course, there's those students that don't have computers, internet access, or a district with that kind of capability. So how do we kind of create this continuum of learning? And in a way, it's almost an opportunity to be liberated. I think as educators of what Tony and I have heard from teachers who work with them, there's a lot of constraints that are placed on the teaching day. This must be taught by this week, and this needs to be done, and these are the tests that are required. And we've seen, except for the really doggedly determined educators, kind of the squeezing out of the creative and imaginative and playful, especially as you get past the grade school. It's like there is no play. Here's the things you need to learn. The test is coming. Grow up, right? And it's like, wow, here's a chance we can actually even reboot our thinking about what all that means. Can we get some questions popping up in kids' minds and make them wonder a little bit more? Liam was on earlier, and I hope he can jump in again. Our snafu this morning, or this afternoon rather, with our media has made it tough for other folks to join in. But I mean, just going out at the night sky, he's really connected with the astronomy community. And next month is astronomers. It's basically it's a month-long program focused on astronomy, and I would hope that he can bring some passionate folks going to talk about it. Just walking out at night and looking up in the sky on a clear night. There's your science, right? Going out in the backyard and getting your fingernails filled with dirt. I mean, that's science, right? How many bugs are in that handful of dirt? You know, how many different plants can you see? What birds do you hear? So I think in a way we've kind of been we've got this tunnel vision of just like pedagogy, like chapter 5, odd questions in the back of the book. It's like, hold on a minute. It's an awesome way to just take this huge pause and reflect in this kind of global crisis about really what is learning, what can we do? And even though we're social distancing, we are social distancing because I'm talking to some dude in Japan right now, right? I mean, that's really incredible. What do we got popped up here? Let's get back to roots and let's talk about what's happening there in the meristem. So who popped that up? Was that you, Michael?
Yeah.
All right, let's dig in.
A little bit of science humor.
Okay, it's hard for me to see it. I'm on this Chromebook with low res, but can I zoom that in? Am I going to make it bigger? Read it out to me left to right. Oh, there you go. Knock, knock. Who's there? Oh, do these. It's perfect. And so this is our physiology. This is what's happening. That hook is an actual hook, isn't it?
Yeah. Yeah.
And the knock, knock, those are the rhizobia.
Right. And then we've got the nod factor. So then the root hair, the gulp, the root hair actually wraps around it. And you get the infection where it the bacteria sends a thread and down into the root and infects individual cells. And then we get the inflammation reaction. And so instead of it being the root hair, it's now the nodule. And this is exactly what we dissected earlier in the program. So
that's wonderful. And so the mechanics of Nod, the land of Nod, what is that's some chemistry going on? Dr. Barker talked about a little bit about this exchange, these chemical pathways that are being established. Is there a little dance going on like old school modems? You know,
yeah, there's the plant itself sends out signals to into the soil to attract the bacteria. The bacteria signals the plant. If it's the compatible species, then the nod factor gets triggered, and we get the nodulation, the infection and the nodulation occurring. You know, the tricky part is remember it's an infection, so it starts out as a pathogen, starts out as any bacteria that we worry about, and then that conversion happens where it becomes symbiotic.
That's a cool little cartoon.
Yeah,
and I think we should definitely make sure that we put that in one of our lesson plans somewhere, if it's not already there.
No, I don't think it is yet, but we'll get it there.
Oh, there's Liam. Oh, and look who he's got with him. Well, he's got the space station. But that's
Camilla's helping us out today.
Awesome.
Excellent.
No. I thought I'd show you Camilla, Michael, just to show you that she's still here.
The hardest working chicken in space.
Liam, apologies for our kind of technical funkiness today.
Oh, no worry. Yeah.
I totally melted down. Tony was really patient with me. Yeah, I have.
I have a
Hopefully
question.
it's all captured.
Yeah, I know, right? He's
YouTube,
got great
right?
editing skills. We're going to need to.
I have a question, so hopefully she's still watching. Lori Waters from Florida asks about the nodules, and she says she's going to do the dissection with her students next week. So Lori, I hope that works out well for you. And her question is it possible to culture the rhizobium from the new root nodules and then apply them to other seedlings? Absolutely, it is. It's really simple to do. You harvest the nodules and crush them into sterile water, and just a few milliliters of that solution, or you can dip your seedlings into them before you plant them. You can actually transfer some of that supernatant to a petri dish and grow your own cultures. And yes, it's not beholden to the science supply company, you can create your own strains. And because those strains have the individual biovars, if you're harvesting from the plants in the environment where you are growing, you're going to have a better strain anyway, because that'll be the one that's suited to what the plants that you're growing. So yeah, definitely give it a go, and let us know how that works out.
Very cool. Yeah, it's so open-ended, really, when we think about learning. What are the outcomes? I think our teachers that are watching these videos, or students, or even the parents here, thinking about some activities for your teenagers, perhaps, or tweens, or even younger. What is appropriate? What's age appropriate? And we can do deep dives, and I think Dr. Barker clearly showed how deep it can go. I think he was just honestly just opening up the hood, because I imagine he can go really, really, really deep if we're ready for that. And then we can just keep it at a really kind of high level. What's photosynthesis, right? And just taking that seed, and Michael shared with us a little earlier, the ability to just take a water bottle and make it our little growth plant with a little wick from just a cotton string. You're all ready to go. You don't even need to go out and get garden soil if you just want to grab out some dirt from the side yard or what have you. Hopefully it's not too clay. Did someone just wake up?
Hey, Gil, I took a picture of you guys.
One thing we might do, there are 34 days in our program that we had scheduled to conclude on April 22nd, but our flight has been moved to October, and it also looks like these recommendations to stay kind of self-quarantined, if you will, or isolation, might be a little longer period of time, that there can be an opportunity to take our homeschoolers or students that have independent thinking and have these sessions. I would like it, led by someone that has that pedagogical practice. Definitely not me. Michael, if you would be up for that, or Renee or Scott or Gil, any of our really any teacher, the 130 some odd teachers that were currently on Flight 7. Whether you're even in South Africa or Kenya or in Quebec or any part here in the United States, we'd love you to showcase your expertise and what you feel most comfortable about sharing. And really, it's any grade band, it's any segment of this. You want to dive more into the bacterium, you want to talk more about the legumes themselves, let's do it. And what we've tried to do in the most simple way possible is say, let's look at a flight we're going to do anyway, and a decision we've already made is to look at these rhizobia in their interaction with legumes and consider what an optimal legume might be for what Gil had brought up earlier. Is it nutrient dense, right? Or are there other factors that your team, your group, might consider beyond that? I wonder who's on Team Clover. Gil, you're going to help me with the Zuki beans, right?
Bro, I got you covered, man.
Awesome.
They're a big
That's
deal
good.
here in Japan.
Yeah.
We're gonna fight for that. Yep.
There you go. I just want to try the bean cakes when you're done.
They're good. They're really good.
Oh yeah, for sure.
Well, I keep seeing that ukulele pop up and down. The two of you going to give us a musical interlude here?
Oh, you don't want to hear my ukulele playing. It's pretty bad. I was given one, and it's like my ADD is just kicking in.
Frozen song with the ukulele.
Oh yeah.
With the ukulele?
Well, we can make it like a science investigation on waves.
Yeah.
You don't have to worry about playing music.
Yeah, maybe your
right?
Azuki beans like different chords.
Well, that's interesting. Actually, Dr. Barker brought that up earlier. He was talking about sound and vibration. He's asked for two instruments indirectly, and I know he'd really love to see this happen. He's a really great instigator of stuff. And one was radiation, and the other one was sound. And then there was a study that was done about how these this trigger mechanism in these plants, when there was a certain frequency that was felt. And I just kind of blurted out, it's probably totally wrong, but the idea that some big herbivore that they just got accustomed to the thump, thump, thump was that right frequency, right? And so they kind of freaked out because, oh my gosh, here comes the monster. He's going to eat my buddies, you know. And so plants, you know, they can smell, I guess they can hear. And, you know, this other stuff, they talk about the trees can talk to each other. I'm way outside of class if I want to walk down there, but if anyone has any knowledge about that, let's talk about it.
No, there's a tremendous amount of chemical signaling, pheromones, and
God.
microbe messengers. We are learning so much more about plants than we ever imagined.
There you go. And here's a shout out to that AP Bio teacher that's watching. I know you tuned in, you're watching this video, and you're wringing your hands thinking, I want to do something. Share something with us, share some knowledge. You know, legume rhizobia is kind of what we're focused on, but broader knowledge and introductions, we welcome that. It's really a chance to build community here during uncertain times, and something we can gather around. Our philosophy, I mean, it's International Space Station, right? 18 countries are actively involved. More than 120 different countries around the world have run experiments up there. We get to do it too. So 5 years old, 25 years old, 95 years old, really doesn't matter the way we were setting up the platform. And so we encourage you to be creative and imagine. Or draw some art for us. You know, those young ladies there, would you draw some art for us? What's your favorite bean or a legume, right? Want to see some pictures. One of our interns from last year, she was in Singapore Sea. She's not with us right now. I guess she's working on our scheduling and what have you. But, you know, she did this animation right when she found out about the program. And it's like our first fan art. It made us feel so good. And so share your art in addition to your intuition about where you might think a good hypothesis will be found. And there are deep tools that we can unlock with folks like Dr. Barker and the knowledge that Michael Wilkinson has, and you go there in Osaka. So, and there's others. Not all of them are here. What can we do? What kind of fun can we have, right? And I apologize for having my video, and I'm going to pop it on just for a second to wave to everybody. Hi, I am here. It's like ISS above. It's my garage. My rockets, I probably won't be able to fly for the rest of the year or the rest of the season. But yeah, so what, yeah, there we go. Here's C's animation. And she told us she had just learned how to use this program. And so she busted it out just because she felt it, right? And the seed in your little agar, here comes your plant, and shaboom, legume challenge. Now, what's interesting here is we have traditionally in the last flight, maybe I'll share that. I'll turn my camera back on just to show the XLR because it really didn't show yet, which is the device that actually goes up into space. Oh, this is in the classroom. What goes into space is more of an aluminum box, right? It's this little guy right here. This is a 10-centimeter cube. Aluminum's got a fan on it. It's our electronics, and our experiment goes inside. A little port here would normally have a connector that plugs into a card. That card gets slipped into a locker on the space station. The door closes. The astronaut closes the door. Boots us all up, and they float away, and they do some other stuff for a month. They come back. They lift up. They pull out the car. They disconnect us. They throw us back in the bag. We go home again. This is a small unit. This is a 1U or 10-centimeter cube. Our exolab mimics a 2U, or you can see about twice the height there. And this is what we have here on Earth. And for this social distancing, we don't really have any equipment that we're asking you to use. All our teachers that are already using it, by all means, go right ahead. You'll find that we do log in. I think our team set it up where you can see our other exolabs around the world if they are hooked up and running these experiments. But really what we want to do is imagine what might be possible. I'm sharing this with you so you kind of understand the home it's going to be in come October in schools around the world. If your team legume is selected, it'll be growing in this kind of device. So the experiment format, will change. So I'm going to take the experiment out here with our plants. Michael's plants were much more mature than ours. And I'm going to show you just XLab and what it looks like. So this is 20 centimeters across here. Imagine like 8 inches long, 4 inches by 4 inches, 10 centimeters. And this is really trying to mimic the same volume that we have up there. It's a different device. It's really just measuring the ambient environment. It's not trying to have a controlled environment. be exactly like space station. That'd be prohibitively expensive. When Tony and I started Magnitude, our goal was to really keep the cost as low as possible, but make the fun as high as possible. So we have an LED array here that the light is adjustable. The intensity of the light, the duration of the light, and even the wavelength can be adjusted. There are presets that correspond to what's happening on the space station. But if you as an educator want to change the light or the intensity or duration, you can do that. We've even enabled the ability to split the lighting if you want to have one side under one lighting condition and one on another. When we run these larger tubes, that's kind of neat. They actually can get different lighting conditions. But underneath that, we have carbon dioxide sensor that's measuring the CO2 in the air, and of course that's nice food for our plant. Little pinhole here has a temperature and humidity sensor, so you know that moisture in the air and how warm or cool it is. And this little bar that's popping out right there is a little PCB, a little computer board with a light sensor on it, measuring micromoles per square meter per second as that light changes and what have you. And it's a measurement there in lux. There's a little port here just for future conversation. Maybe if we have any engineers that are really excited about devices like Exolab, this port could be an expansion port to add maybe another instrument down below here in this little back area. So maybe different gases you might want to measure. Again, this would be ambient. Maybe it's something like a different imaging method beyond the human visible light spectrum, infrared or some kind of spectroscopy. How about microscopy? Could we fit a lens in here that allowed us to zoom in a little bit so we can see things in much more detail? Our platform actually does a fairly good job of the images where you can select them, they zoom up pretty large, but maybe you want something even a little better. So that's Exolab. It runs a little Raspberry Pi on the back plane with a Wi-Fi connection. So it connects to a local network, and boom, all the information is going up there. Oh, it also has a camera. Oh, it's kind of hard to see it in my lighting here. There it is. Fisheye lens. And Dr. Barker's also suggested there is MATLAB has some software we can run to actually clean up that curvature that we're seeing right now through this fisheye lens to give us a more accurate reading. Him being the he wants to quantify everything he sees or all the information he's receiving, so he wants to really get accuracy. So hopefully he'll help us out with that, so our imaging is going to start looking more true rather than that wide angle, which would be kind of cool. The Exolab captures an image once an hour and sends it up to the cloud, and then we can kind of compare what's happening in classrooms around the world with what's happening on the space station. So that's pretty neat. So that's Exolab, and that is kind of like the star of the show. Our first mission went up in 2017 in February, SpaceX mission out of Kennedy. And we grow different, use different form factors. And a consideration when you're thinking about your Leguminaut that you want to fly is maybe you actually want to use a different vessel or even perhaps a different media to grow. And we're open and welcoming your creative insights, your keen observations, your quantified reasoning why we should be looking at doing something different. And so I challenge everyone, teachers, student alike, other professionals that might be joining us saying, hey guys, if you did this, it'd be a lot better. Because, you know, raising the fidelity is something we want to do. And through engineering exploration, sure, why not? The current mission had these larger tubes here on Earth, and there were smaller tubes. Michael, what was the dimension again on orbit? We fit, I think, 3 across.
On orbit,
but
they were
we also have
25 by 130.
There you go. So they were a little smaller. We've had smaller tubes in which you've flown the microgreens, so we could fly 4 across. We were looking at, you know, succulents and other types of plants that would be edible. And of course, we grew multiple so we could get the most bang for our buck, if you will. It's not cheap to fly up there. By sharing what we're doing with as many teachers and students and schools and districts as we can around the world, it dramatically lowers the cost to be able to do this. Several orders of magnitude. How about that? That's why we called ourselves Magnitude, right? So that's, let's think about vessels. Anyone please weigh in on thoughts on that or on media. I know, Michael, you have your favorite media. You took your plant out today before you went into your dissections with just your probably some good soil. well-balanced nutrients, but you also use arsenite, I believe.
Vermiculite, same
Vermiculite.
basic. Yeah,
Yeah.
it's mica that's
So
been
let's
baked
talk a little
and
about.
it puffs up, and so it's very light, friable. It doesn't compact. There's no nutrient value in it, so you're going to have to provide the nutrients that the plants need. In this case, the potassium and phosphates. They'll be fine producing their own nitrogen, or gaining their own nitrogen if you have the rhizobia in there with them. But just your typical Osmocote or Miracle Grow, something like that, you can mix into the water, and that does fine. But for the short duration on these, I've had good luck without actually fortifying the water at all. So it's
now let's talk a little bit maybe about the agar, the gel that we've been using, and what is that all about, and why do we use that? I mean, why not just use vermiculite or dirt, soil? Or can we do something cool like use lunar dust?
Here's a good thing to think about. Why aren't we using? Where did I put? Oh, let me put the cap on this first. So this is the agar. That's what's going to happen to this in microgravity. That's gonna be bad news. The astronauts are not gonna like little bits of vermiculite flying around floating around the environment.
I bet the Millennium Falcon can fly through that with no problem, though, right?
Now, there are groups that are experimenting with media like this, but they're putting it in a pouch so that it can't become FOD, floating orbital debris, or foreign object debris. But the agar stays, and it's got all the nutrients and the water that the plant needs for the short duration of our flights, which are around a month. But after about a month, they do start to
like desiccate,
use up their moisture. I was just looking for one. It was pretty amazing how much was gone.
It is like the plant's roots are like straws.
Yeah.
And the edges of the tubes. But we have, the formulation of that agar is that we use a relatively standard formula. We can share that with you guys. Murashige and Skoog is the formulation, but you might want to just try an agar, just get some gelatin, make your own. That might be an interesting way to get started. Maybe you want to just plant yours in soil. And really, even if you didn't have legumes, and you maybe, I'm hoping you can get some seeds somewhere fairly easily, just to try these out. There you go.
So you can see that this is really the moisture's been sucked out of it. But yeah, look at that root system.
He really wants to go. Which plant is that?
This is a cowpea, and it's an experimental one.
Okay.
So hopefully we'll get some nodulation out of that one.
Well, there you
go. I apologize. My family and I got to run.
Okay. Thank you for joining us.
Yeah, thank you.
Thank you so much for having me. It's been great meeting with you guys. It's super encouraging. Just if I could leave on a positive note, this has been a really testing time for everyone around the world, but it's kind of brought to the surface things that are really important, both in our careers, like what is essential, what is necessary. It's brought creativity that we're able to kind of see through the fog of our daily routines to the things that are needed, and kind of at a personal note, our family, we've been able to kind of realize what are the things that we haven't been paying attention to on our day-to-day because of how busy we've been with normal life. So let's try to look at this quarantine and this pandemic through that lens as to what can we grow through, what can we develop as a result of this. Because once this kind of calms down, we're going to kind of return to life as normal, but it won't be the same, and it shouldn't be. So taking the lessons that we learned from this experience and growing further into a better standard of operations, if we can say it that way.
Right. That's a wonderful way to be thinking about it, Gil. Thank you so much. And well, let's set up a time with you, me, and Tony, Michael, and we can talk a little bit about the Osaka, Japan program and what we're doing here in the States. And really, although you're now kind of on the opposite edges of the world, what we can do in Europe and in Africa and really kind of light the fire. I think if anything, our social distancing has made it distant, and we're connecting those distances, and it's really, really pretty cool. So thanks for hanging out with us again, and it's great to meet your gals. And you're going to draw us a picture, I hope, right?
Girls, can you draw some pictures of plants in space for them? Can you do that? You want to do that today?
That would be so awesome.
Yeah.
Thank you so much.
Right on.
Awesome. Have a great weekend, man. And
all right, guys, let's go. Right on.
That's right.
All right, see you later.
Thanks, Gil.
Bye, Gil.
Now, Michael, why don't you just what's Camilla's backstory there?
So Camilla started out life as the official mascot for NASA's SDO mission, Solar Dynamics Observatory, and her purpose in life was really as an ambassador for that mission. Tweet on Twitter and Facebook and Instagram.
And what's her?
She's
her
at Camilla Space on Twitter and Camilla Corona on Facebook.
Camilla Corona, really?
Yeah, Camilla Corona.
Yeah, I've done a really bad job of representing her.
But her main purpose in life is really to get people engaged in STEAM activities, and she's 16 years old, so she's a little snarky and can relate well to kids, but adults also seem to love her, and whenever they see her, they always want to get a selfie with her and tweet it out. But this is so with a couple of furloughs ago with NASA, she got excess, so now she's a free range chicken.
Wow. I wonder what a fertile egg would do in microgravity.
I don't know.
Is it has it been done?
Not poultry, but probably fish. I wonder if the Medaka, did those get sent up as eggs, or were those already?
I was under the assumption they were already done, but that was a really interesting study about how fish understood up or down, and they're already in a three-dimensional space, right?
Yeah.
But they did get a little confused, I guess, which was kind of interesting. Kind of like those the roots that Dr. Barker was sharing with us yesterday in the darkness and in microgravity.
Yeah, I mean, from a physics standpoint, gravity is a weak force, but it's amazing how much of an impact that weak force has when you take it away.
I think it's a bunch of little weak forces adding up to a pretty massive, impenetrable thing that's very difficult to get away from, unless you're traveling at so many meters per second. So let's see, where are we? We've still got another hour here of engagement. At least we promised to chat about that. We can loop back around again for those that are just joining us now. I really love your mission patch, Michael, by the way, the Exolab there on your arm. We all need to get suits. But can you share again what you had done just to show up and talk about the process? How long did it take for those nodules to develop, for example? And why might they not develop? I guess would be another question.
Okay, sure. So the bacteria, rhizobia, and the legumes have a symbiotic relationship if everything goes as according to plan. The plant itself sends out chemical signals into the soil that attract the rhizobia, and the rhizobia are then actually captured by one of the root hairs, and it just hooks around it. And when that happens, the rhizobia infect the root system of the plant, changing the physiology of some of the cells there's actually a reaction that creates these little nodules. And earlier in the broadcast, we dissected some. We harvested a couple of nodules from the roots of a healthy legume, and it's not looking too bad for the transplant and having a little bit of a rootectomy. I'm sure if we looked at the chemical signals that Richard was talking about, it's probably screaming at us right now. But
fluorescence and a little GFP on that plant before we did all that would have been kind of cool.
Yeah. So that process starts, as I said, with the root hair. So at the seedling, and then that all develops with the plant. And that's the little factory, if you will, where the atmospheric nitrogen is broken down. It's, you get N2, you got two nitrogen molecules, and I was just talking about weak forces and strong forces. That's a strong force, and that force is so strong, there's nothing that can break that apart except a lightning strike. And these little bacteria, they're able to break those bonds and recombine the nitrogen to produce ammonia, and convert that ammonia into nitrates and nitrites, and that's what the plants use. That's what we think of as fertilizer. And those nitrogen compounds are critical to life because that's what DNA is made of. That's what proteins are made of. And if we don't have those compounds moving through the environment, we're not going to have the ability to sustain any life. Now that plant that I just pulled out, I was showing you one in a test tube, and that was kind of so we've got these that are growing in the test tubes like they are in the ExoLab. And while we've got a nice healthy plant, and this one probably is a candidate for transplant soon. I've been transplanting them so that we could actually harvest some of the seeds, and do some further experimenting with them in my class, and also be able to hopefully have enough that we could make a little dish of cowpeas, something that we could share and taste. So we've got this garden. I quickly grabbed all the plants the night that I heard we were closing down and brought them home and set up my greenhouse here. But I've also got a whole collection of them up in the living room on the windowsill. So you don't have to have all the technology to do this. You just need a good source of light.
Oh, there you go. A little water.
That's about 5 weeks old. The first nodulation, you get pre-nodulation in about 3 to 4 weeks, and those active nodules like we saw, where they got the red or pink center, that takes another week or two, and by the time you've got the plant flowering, you should definitely have very active nodules.
We scrambled to put something together here over the last week, and this challenge has a framework, but I think it's still loosely defined. We understand what we want to be able to do, down select to a flight candidate, a legume that will fly with a specific rhizobia to have a successful mission. But in the hour or so that we have, why don't we together here learn about some of these different legumes that we've kind of picked up as candidates. Now, I'm going to confess that I know very little about any of these, but I think together, even here, we can learn and kind of think about what some of the criteria are that we're considering. Now, if Tony's still with us here, Tony, I know that C had prepared this beautiful spreadsheet that began looking at the percent of carbohydrate and the percent of protein and some others. And I think that's an interesting lens to look through, right? Another one is might be how much water does it use, or is it relatively a teetotaler and is sipping just a little bit, not really consuming a lot. And so I think that's a consideration. Are there any medicinal benefits, or if it has a long history with human civilization, why was it that the plant, the one that kind of evolved with agriculture and the little villages and turned into towns and what have you? And so there are a lot of different ways we kind of explore there. just through any of these given personalities. So shall we do a deep dive and grab a lagoon and just kind of investigate one as kind of like, here's a little snapshot? And maybe if that's a format, when we pick it up again next week, we can kind of do that and have a little showcase. And we're not professing that we have all the deep knowledge. We're really kind of just opening the door here, saying, here's what we need to do. Here's our basic objective. And I hope that you as teachers, parents perhaps, are definitely our students, it's open-ended for you to kind of begin looking at and investigating. Something Dr. Barker said yesterday, treasure hunt, when he was talking about what drove him and gave him that the motivation to move forward in his research and his investigation. It was to make a sense and find a pattern in something. But we don't necessarily need to go down that route. We can if we want to with data science and being able to quantify and measure the chemicals and the changes in reactions. But we also can kind of pull it back a little bit and think in a more broad sense in terms of what triggers us. So let's grab one of our lagoons perhaps and do an exercise for fun and just collectively here brainstorm. Scott, Renee, Tony, Michael, and anyone else that wants to chime in. Maybe you can do this at home, right? So should we spin the big wheel? I need a wheel here. I can spin, and we get, right? Do some randomizer. So what do we pick? What's that list? I don't. It's going to be hard for me to connect here with this Chromebook, but if we can pull up our page, Tony, if you're there, and maybe show our list of legumes. Awesome. Oh, this is great. So this is what I was talking about, and thank you, Tony. And so what we can see there is that we started to fill this in. Don't wait for us, right? If you want to do some research here, what sounds kind of interesting to you? What
Looks
maybe
like
you
Team Mundy's been busy.
Yeah. So we have some leaders. We've had at least one person assigned to each lagoon, and you heard me a little earlier kind of asking Gil to help me out with zucchini beans. And so I guess we're gonna need to populate that. You might actually be thinking these are the columns with each of the legumes that we're considering. Maybe there's an additional column you want to add. Scott, I think you mentioned peanuts, right? It's not a nut, it's actually a legume. So if you really want to do that, Scott, let's make a column and say peanut, and let's talk about why we'd want to kind of consider that versus something else. Why not? So it doesn't need to be stuck in this framework. We will need to down select. And so you need to kind of find other people to support your cause, right? Draw a picture, write a story, do some science, do some testing. Maybe if you want to look across the rows, you're most really keen, super excited about B12 or whatever. Look across it, maybe help us fill that in and say collectively, I'm really interested in B12, and this looks like this legume does the best job, and that's why it's the plant we want to use, because B12 does X, Y, and Z. So there's a lot of different ways to kind of look at the challenge, and there isn't any one way to do it. And so please, if you've got ideas, post them in our channel here in discussions, send us an email, comments, or add us as a friend on Facebook on the Magnitude page. Let's have a conversation. Let's think about it. If you're a little at odds about how to migrate what you've done traditionally in the classroom now, you're not looking at your students face to face and they're remote, and some of them you can't even get to them. How do we solve that, right? And so feel free to reach out to us. We're locked down, right? I am for sure. And I am excited and interested in hearing from everyone that's watching these to learn what you're going through that's different than what you have been doing, and how are you adjusting? How are you making it where you're in your comfort zone, or best as can be. I know we're dealing with our own personal families and our own concerns, our needed neighborhoods and communities. And then as you as educators, you're trying to deal with your administration, your students themselves, their parents. And I'm going to guess there's more than one helicopter parent that has been filling your inbox with, what's Susie's lesson for today, right? It's like, and how do you manage that, right? So this is the things that I think I would love to hear just as an entrepreneur trying to solve the needs of learning in the 21st century, to get people excited and get students excited. So let's drill into one of these. On our homepage, we actually have some beautiful images, or we just do a random here. What do we have now? 3, 6, 9, 1 of 9. Yes, Renee.
So I actually have to sign off. I think what I'm going to do with this is, since I am taking alfalfa, I know a little bit about it, is actually try to engage some of the homeschool families and reach out to some that were interested. But I think we're a bit fearful to get involved with this, because especially with the lessons, and especially for some of the audiences, I think what we're looking for here, have them get involved with even the forefront, and gather information that way, because I think that'll be a great way for them to do some research and find out information to help me populate this field. So it's not just me, it's me engaging, because we have a lot of homeschool families, and a lot did express interest, but then they couldn't do it. So I think that that's why I'm going to take it and sign off from there, if that sounds good to everyone. To
Renee,
Tony.
that would be awesome. And also, if you want to chat sometime next week at a time that's convenient for you, anytime after 7 a.m. Pacific, maybe we want to brainstorm a little bit. We had talked about doing some type of teleconferencing for the homeschoolers that want to do that. Let's set up a little session just for you guys. That'd be fun.
I think that'll be great. Yeah, because like I said, especially, and I think, because there's so many changes right now, I think it's going to be tough from the district side. But I think with sort of the homeschool, and especially that's going, I want to keep that momentum and find new ways to keep them engaged as well, and find ways maybe to support some families that are looking for other things, that have interest in the space station, and, in a different way, to get them involved. And I know we talked about some of that with the public engagement piece with this, because I think we're going to see more families looking to do more of some of these things, and even a smaller scale. So I'd be interested to see how we can, just help get people excited and have something they can focus on that's interesting and different. So.
Awesome. Well, we're here to help you out any way we can.
All right. Thank you
be
so
your
much.
wingman. All right. Thank you. Take care. Be well. Have
Bye.
a great weekend, huh?
Oh, you too.
All right. We'll see you, Renee.
Bye.
So, yeah, of these, let's pick one.
We could go and look at our cowpeas here.
So that's one we flew, absolutely. So I'm going to go ahead and just see what I can find about them. Homework. For you, those listening, if you're actually dialed in, you want to do a quick little search. We might actually even want to think about this as a format next week. What do you think, Michael?
Yeah.
And those that are hanging in, so we do joint research and we share our findings and what have you.
Yes, definitely.
Creating some structure. We have this kind of general idea, and there is no structure, but I think people, especially now with everything so unstructured, maybe we're going to kind of structure a little bit. So cowpea originally originated in Africa, right?
Yeah.
That ancient West African cereal farming. 1,000 years ago. Wow.
Hey, Scott, do you want access to this spreadsheet?
Yeah, if you can just, either, put it, either email it to me or just put it somewhere where I can download. That'd be great.
Okay.
You probably should, when you're ready, put it, put up the blank one. I would say, or not, but I would say use the blank one, because then you can divide the responsibilities in groups of kids. I think that most of this stuff, you could assign it in teams of 3 or 4.
Love it.
And rather than have a whole class research one, because if you do that, really what you end up doing is either you'll have one or 2 kids do the whole thing, or it'll just be every kid does one line in here. There'll be some discussion, but I think it would be better if you do it where you have all the legumes and have 3 kids focus on, as a team, on one legume. Okay, although, that works obviously a lot better when you're in the classroom than right now when the kids are all separated and you don't know when they're going to get together. But I think it would still work better that way.
Sure, but I mean, even with the Google Doc, that can all be done asynchronously too.
Yeah.
So
the key, and yes, that's true that the kids can. I do almost everything on Google Docs for precisely that reason.
No, we can totally.
I was thinking of one thing. If you're talking about the 8th grade standards, we don't do a lot with plant structures. We don't, although 7th grade, this would also work very well, because we do a lot with environments and not environments and ecosystems, and therefore talking about symbiotic relationships. This is a perfect example. But also, if you're talking the 8th grade standards, you're mostly talking about heredity, and you're talking about adaptations, and you're talking about genetic modification. One thing you may want to talk about here is, although obviously the leguminate we send up will be a specific strain of legume, talk about what genetic modification could be possible when you're talking about, since we're talking really about what would be an ideal legume for, farming in space. Start talking about what modifications, because you're talking about soybeans, they've been modified to death. The number one GMO you're probably eating is soybeans.
Mm-hmm.
But what modifications could be made to make them more suitable for space, to increase their nutrient density, as you were talking about? Or if you could do a play like you do with golden rice, and suddenly, you can get your B12 that way, or get a vitamin that you wouldn't normally get from that. Is it a possibility?
Oh, that's very interesting. Getting into a little kind of genetics here. And one thing that I really liked, Michael showing us that physicality of getting the students involved in an activity where they can kind of just be out there in their yard, or side yard, or what have you. How do we get seeds in the hands of students, right? Not every student will have a laptop. Do you know if all your students have the capability of getting online? Scott, are you fortunate enough?
No, there are kids, that don't have the access that's needed. Now, that being said, I'm trying to make it very public, and everyone should, that I know at least Spectrum is offering for people who don't have internet access 2 months free.
Okay.
And another big thing is a lot of the people who do, the programmers who are doing the online education services, for fees, are basically, suspending those fees, and at least a lot of them. So it's a great time if you want to try out all these programs that you wanted to try out, but you only had limited access because, everything's, a little huge chunk of money from a district that doesn't have that much money to spend. Suddenly you can try everything out for real, rather than having 1 or 5 practice accounts. So
well, you've turned that into a benefit.
Yeah.
This is your extended trial period.
So it's,
how many things have you downloaded?
If you want, this is the time to, and also because all of a sudden, yes, you can experiment more, because you've got to try different things. Suddenly you're not chained to the book.
Being able to stay in those standard frameworks, though, I think is helpful for the students in the long haul, as you're kind of advising. And that guidance there, perhaps, if you look at it as helping us create a scaffolding for other 8th grade teachers, if we're thinking about the next generation science standards, that could be helpful. Anyone that's on these channels that's really passionate about standards and wanting to make sure students have this kind of foundational learning can maybe help, with 3rd grade, 11th grade, whatever it might be, help build around it. There's so many different lenses we can look at through this program of putting something into space. It's not just the sciences, right? You got history, and you've got language arts, and there's just so many different ways you can look at this if you just kind of open your mind a little bit and think about, how can I do it differently, since it is all different right now, right? If we have a hammer, everything looks like a nail. And if we're stuck in a routine, then we're basically looking for those nails. And now, well, there's no more hammer, so maybe you still want to look for nails, but maybe there's something else we can do. And that's up to each one of us. We're excited to see what happens and what goes on. So looking at CalP, why don't we fill this out now, since I've kind of committed ourselves to doing it the rest of this video, and we'll use it as a guide and just walk through it kind of extemporaneously here, and imagine how you might treat this in your class, whether you do a blank spreadsheet like what Scott's recommending, or whether you're waiting for it to get all filled out so you've got a format to then introduce to your students. Really kind of up to you, no one way. So what else are we learning about? Here I got, I don't have direct access to that. So if you pop in protein, 24.8%, fat, 1.9%. So 1/4 of that CalP is protein. Very nice. And then fiber, 6.3%. I guess I need to got that written down, but I don't know if anyone, whoever's sharing that sheet, if they can type that in.
Can you read it to me again, Ted?
Sure. Protein 24.8.
24.8.
Yep. And it's hard for me to read the screen there, but I've got
Fiber.
fat, fiber at 6.3.
Sugars.
Sugars. I don't really have anything showing sugars on this one. We'll leave that blank now. I do have carbohydrates.
Yeah.
63.6. This is according to Purdue, by the way. I guess I should source my numbers. Fiber, the calories. I don't have a calorie count here. These are
vitamins.
I'm sorry?
Vitamins.
I have thiamine, riboflavin, and niacin. That's the only thing I'm showing here. And these are fractional percentages. So those primary things, I think, here's some just text here. Again, this is from Purdue.edu. This is New Crop, and I guess with as a source, I'll put this in a, looks like it's their horticulture department. I keep on trying to do my Mac commands on my Chromebook. Let's see. It's rich in amino acids, lysine and tryptophan. Isn't that in turkeys?
Mm-hmm.
Yep.
That's interesting. So you get if you eat a lot of black-eyed peas, do you get the same feeling?
I don't know. I have to pay attention to it.
No, and I'm going to really kind of destroy these names. Methylylene and cysteine. They're deficient in that compared to animal proteins. So
yeah, those are the amino acids. So that's the thing. I think or in the earlier broadcast today, you were talking about the three sisters.
Mm-hmm.
So that's one of the issues with plant proteins is there aren't very many plant products that are a complete protein, meaning it has all the amino acids.
Mm-hmm.
And one of the ways that you can get that is by eating combinations of plant foods together. The three sisters, the indigenous peoples of North America would plant corn, bean, and squash together. And when those are consumed together, they often made like a succotash, and literally we're eating them mixed together. But they're, those three plants actually do provide you with a complete protein. The complete proteins, quinoa is a complete protein, and there's another, amaranth might be a complete protein. So there's not a lot of them that are. So it is important if you're relying totally on plant-based foods to know which are deficient in which of those amino acids.
Well, there's the ultimate challenge for you. With just looking at the legumes, could one configure a complete amino acid balance? I mean, you basically cover the whole spectrum. There's going to be some deficiencies somewhere, maybe an overabundance other places, but is it kind of a Tetris? Can you kind of line these all up and get it all to fit well? And I don't know, again, hard for me to see this the small numbers here on the spreadsheet, but do we have amino acids?
We don't have that as a category, but I think that
that's a great conversation. If anyone's joining us here and listening to my meltdown in the beginning, and hopefully a little more mellow here now in the back end of this conference, we need some help. So you got some insight on amino acids? Share them. Maybe what we need to do is find a way to get this spreadsheet out there as a download or what have you. It can just be a Google Doc or a blank Google Doc. People can kind of copy and use it as their own. I don't know, Tony, what you think about that. That would be kind of nice. We can aggregate them on our site as a kind of a master list.
I don't know if Tony's there. You can scroll down. So what I'm doing further down is putting the amino acids, and then we can put either strength or weakness.
Great. We were talking about having some fun, just kind of imagining what this might look like. And we started coming up with these little personalities, these characters, right, of what each of these legumes was capable of doing. And so maybe it's some fun. You want to get a little creative, guys, students and teachers out there. If you were going to make any of these, give them a little personification of their attributes, what would they be? super strong, or nice and chubby and slow, or right, super fast and quick, and are they wistful, or are they like tough? There's a lot of different ways to kind of think about that. And that can be playful and might help the students get engaged and learn and think about things in a little different way. Let's not try to make it feel like school, I guess. If I was right in the sixth grade or ninth grade or something, I'd imagine I'd probably be doing the most I could to not do homework, to not. Hey, man, the bell didn't ring. So what do we do to kind of get them hooked in and excited and interested? Do we do maybe, Tony helped me with this, do one of our collaborations, but really more ad hoc, in which we call for anyone that wants to register and convene an international hello, and not really necessarily talk about our experiment, but just connect with everyone across the planet that's interested in space, in the space station, in what we're doing with plants and biology or other interests, and just see what we can do to connect. And then maybe hear, like we heard earlier from Scott, Renee, and Gil, about teacher experiences. And Michael, my goodness, you've got some stories and insight with respect to both your adult children who are at university now, dealing with that, and also your young ones that you educate, you teach in school, and that dynamic and how you're managing it. So in a way, it's also these sessions are designed for us to have a chance where we can find a community and a common place to share our frustrations and or concerns. And if we can stay focused on a mission or a project, it's a little easier to not that's something I think we can control a lot better than whether or not our neighbor or our relative or we ourselves might come down with this virus. So I think if we can direct ourselves into actionable, it makes it a lot easier to manage. And if anything, I think that's been our impetus for setting this up. Say, hey, we're here. Kind of appreciate what you guys do as teachers and students. It's not break. Hopefully you're still learning when you're my age, late middle age. You never stop learning. That certificate you hang on the wall, say you graduated from high school or that you graduated from some college or university, it doesn't end there. You've been given the tools to go out and to continue learning and applying those skills in something you love to do, or something you feel passionate about, or something else that really motivates you to get up every morning. So, it doesn't stop after you pull that tassel from one side of the hat to the other, that miter board. Right. And we're here hopefully to do some inspiration, not drone on and on too much, which I'm doing, so I'm gonna shut up now, Michael.
Well, they call it commencement, not culmination.
Right. Commence to what? Right. So, hey, you know what? Maybe let's provide a little certificate of participation. That's something we haven't talked about, right? So, teachers, I think that reward loop, intrinsic motivators have helped a lot of people out. Some folks like me are a little more inclined to be intrinsically motivated. But let's think about some extrinsic motivators. There's some actually really cool things out there, like Badger, spun off from Mozilla Foundation, in which there's a digital badge that shows that you've had an achievement. So maybe you've successfully planted a seed and watched it germinate. Oh, you've unlocked a badge. And what's cool about Badger, it's open source, and it has a digital record. So if you have a little badge that says you just learned how to fly an airplane, it will link back to, maybe, I'm making all this up, back to AOPA, the Association of Pilots, and that it authenticates that badge and says, indeed, they are a pilot. They can fly an airplane. Or someone can put that badge up and it's like they click through and it's like, no, you just kind of snag that badge. It's not authentic. You really aren't a pilot. And my goodness, I'm not going to a plane with you. Or you don't have that level. So continuing education, achievements for students, variable reward loops are a real thing. Vegas is based on that. Give them an immediate reward and kind of then give them another reward, but you don't know the timing. That's how one-armed bandit gets pulled day in and day out, because there is that reward, easy at the beginning, and then it gets more difficult as it goes on. Probabilities, right? Can we input that into the learning system, learning process? We don't have the energy or effort to go overboard with that right now. Right now, we really want to create a safety net, a psychological safety net for all our educators and teachers out there, and a fun project to explore and do. But these are ideas that if you have ideas like that, we love to hear. We're a small company, and I think we're pretty agile. I'm really fortunate to have a really amazing team, really good people. And if that idea resonates enough with the realities of what's happening in the world, let's go do it. We'll put it on our game plan, and maybe for fall of 2020, we can see some inkling of that idea that you had that we can use. So this is the chance, right? It's no longer the same game. The bell's not ringing every 50 minutes or whatever, but you've got classes in session. How can we help you make that class what you envision a class to be? Here's the chance to kind of recraft it, put it back in the forge. This is the crucible where you're going to have an opportunity to bring out of that forge this new thing that you've always had in the back of your mind. If I only had a chance to do X, well, X is sitting right in front of us right now, and grab it, right? Go ahead and do it. And what's nice is you don't need to get overburdened by, at least as far as I can tell from most parts, with the process of assessment. And we do want to see that they're engaged, that they're understanding. Perhaps you might want to move in that direction. But for right now, I think families and children, they're looking for peace of mind and stability. They also want to play with their friends, and they can't. And so what can we do? And I know what we do at Magnitude is just a tiny little part of all the super cool things that are out there, not just in space, but just around the world. And I'm hoping to hear from others that are doing cool things. I'm seeing a lot of cool stuff in my feed. But I do want to stress that we are really, really lucky to be part of the Space Station Explorers. And if you go to spacestationexplorers.org, there is just a ton of content there across all grade bands. You'll see Exolab there and Magnitude and the work that we do, but you'll also see story time in space. So for your young ones, let's have an astronaut read them a story. Well, that's pretty cool. If you have that book at home and they can read along, that's really neat. Maybe it's looking at some experiments from Genes in Space, or in a past experiment and the data is available. There's a camera that takes a picture of Earth over a given location. I'm not sure if that's open right now. I've got to look, but that is a really amazing program from the Sally Ride Foundation. I originally, I think, supported that. I'm not sure if they still do, but I do know that is EarthCam, EarthCam, KAM. That's on Space Station Explorers. That's so cool. When it is open, you basically go around the world. You look at the flybys of the International Space Station. The ISS above, I see Liam still with us here, gives a kind of a view where the space station is right now with a live view of Earth, but also where it's going to be and where it's been, at least over the next orbit. And EarthCam lets you kind of position several weeks, I believe, in advance, if not even months in advance, to guess. And the farther you go out, the less likely it's going to be accurate. But you could say, I want a picture on this Tuesday over Kilauea, right, in Hawaii, because I want to see what it looks like right now. And when the space station flies overhead and EarthCam knows that's the longitude and latitude, shaboom, there's a camera that's taking a picture for you. There, you're in Piscataway, New Jersey, or you're in Trenton, you're in Iowa somewhere, or you're in Seattle, Washington, and it's taking the picture of what you want to see. Maybe it's your house, maybe it's your favorite park, and so that's fun. So there's all these cool activities that are out there. We're doing our best to just kind of get it going. As we look over the next month or so, I think it's probably going to be longer now if we can actually shoulder that. I'm expecting to see other folks join us in a more full calendar of experiences, so you don't have to listen to me drone on and on, getting kind of excited and passionate about finding a solution to a dilemma I feel that needs a solution, right? Making it easier, not more burdensome for teachers in a different environment than what they've been trained and practiced for, to feel comfort and confidence that they're providing what they want to do for their students, that knowledge transfer, that excitement, the core discipline, whatever it is that you feel as a teacher they want to have. I'm excited that we are given this chance. As long as I can keep my cool and get all of my equipment working, it's going to be a really fun ride to learn from everyone. And If you want to jump in on the direct inside the Zoom here, you might pick this up on YouTube. You might have found it on our Facebook feed. Send an email out to team@magnitude.io. Tell a little story. Tell us where you're coming from and what it's about. You might be a high school student and you're really looking at writing a paper on this and you've been researching it. And so, man, come on, let's find some colleagues of yours and do something kind of cool like that. You're a teacher, you're looking at doing something, and you want to share it with the network of people that we're connecting. Send us a little note, team@magnitude.io. We'll pick up the phone. We'll do a little Zoom together. We'll find a way to make that fit. Everyone has something to share. I'm just trying to get it started. I know that it's successful when you don't hear from me anymore. I'm just a little icon in the corner, listening to all the cool things you guys are doing. So for right now, my job is bang the drum and get as many people in here excited as possible. And Michael, thank you so much for taking a leadership role here and taking us through this investigation. Flight 7 just now concluded, packaged up now in a flight bag, heading home. We'll be back on Earth here on April 10th. Our next flight on Exolab 8, the SpaceX 21, has been moved from August to October. And that's nice. It gives us a little more time to think about our Leguminauts and what we're going to put in them. A little chance to do a little more research. If we can get seeds out to different interested parties, maybe we can find a way to do that. I'm not quite sure about all the logistics on any of those things, but it'll be driven primarily by your interest and passion that you bring to what you see on these teleconferences. Scott, thanks for popping in and being a rock here during the uncertain times, and really would love to find a way to connect with your district. I know there's you guys have just gone through the dynamic changes, and so reach out to me and Tony and to Michael, and let's figure out what we can do to make sure that Mike, I know you and Mike work real close together, but your elementary school teachers, I think they might be a little frustrated right now about what's possible and what they can do, and but I don't have my Exolab with me. You don't need it, right? It's okay. But my kids don't have computer access. That's okay. Can you see us? Fantastic. Let's give you some ideas. One thing we had was the idea that these lunch services, these breakfast services are going out. Maybe districts can actually put together a little PDF, crossword puzzle, facts of the day, interesting homework assignment. Hopefully it's not that, but maybe it is. But the idea is like there is a distribution platform that's already kind of being put in place. It's physical. And for those students that don't have internet access, don't have a computer, are difficult to reach, they're probably a family in need anyway, and so they're probably part of a food program. Now, not all families are actually going to be able to get to those drive-through food lines, and so that's even a greater concern. Again, if we can, I'd like to get 100%. We can get in our 90s as far as reach in a district with students across the United States, 50.1 million students. I think it's an opportunity, but I need your help, teachers. We need your help.
Yeah. How great would it be if the school put together a little kit with some seeds and some vermiculite and a water bottle, send it home, and have the kids assemble their growth system.
So here's what we can do, and here's what we can't do. I don't think we can solve everyone's needs to have supplies out there. But I think with a concerted effort, we can probably write a nice little document up, have it stamped by some officials and some really good friends on the National Laboratory and the Space Station, saying, here's why you want to do it. Maybe have a list of simple materials that can be used. And let's float that to your assistant superintendent and your teachers on special assignment and your curriculum directors and say, guys, all we need is $2 worth of materials for our kids, and this is what we can do. We can plant stuff in their backyard, or we can do observations, or what have you. And you know what? Let's do it. Let's figure out a way to make it happen. I understand they are holding back on the testing. So, if you need us to write a letter of support and maybe even go bigger and get other letters of support from some larger folks, maybe what we need to do is let's get something like that together, make that available where you can download and say, hey, here's what we're doing. But for now, this is what we've got set up. We can't boil the ocean here as far as solving all the problems, but I think this is the most immediate way we felt Magnitude could serve its purpose in the learning process. And I see Liam. Hi, Liam. Liam, how does that change you? You love where you are in Southern California to get out in your car and visit schools, be engaged, do astronaut contacts where they're talking to the students directly. that really, like, what's your world feel like? You know, all your astronomy buddies that get together and for night observations. You're on mute there, I think.
Muted. Wouldn't it?
Oh, there we go.
Help if I unmuted. Yeah. So, yeah, my you know, life has been turned upside down as everybody else's, and so this is a great way to fill in on that, and I hope to do more of it. I heard you mention something about the Global Astronomy Month program that's coming up with Astronomers Without Borders. That I'll be supporting, and you know, that's a program that usually is based around having communities all around the world conduct star parties, and everyone gets to share in this worldwide thing about what happens when people look up and experience the night skies. And it's a way of connecting people together. Now, obviously, you know, in this current worldwide pandemic, that can't be done in that way. However, it's not standing in the way of looking for other ways to provide that type of community outreach. And so on April 1st, there will be the launch of Global Astronomy Month. There will be a Facebook Live. I should be producing that for them.
You need to produce ours, Liam.
Sorry. Yeah. Anyway, you're doing a great job. I know there's always some technical things that show up every now and again, but the point is you're here. So yeah, so that's what I'm doing in those ways, and yeah, so it's a new normal for some time, and I'm committed, like you are, to provide a space where we can provide that type of connection and outreach, and really engage people beyond the coronavirus. And this is really great what you're doing, and you know, hopefully we can have a you know, this community grow over the next 35 days. Although, yeah, it sounded like that was new news, right, Ted, that your flight has been delayed.
Yeah, Diane Matthews sent this little note saying, "Oh, thank you so much for it, and just so you know." So we didn't get the latest in-flight schedule, so we're going to have to get that and see where we are with our calendar for 2021 too.
Right.
Yeah. You know what would be interesting? We don't really have a format. We've just been kind of rolling through it and just figuring out, you know, telethon type, we're here for you type thing. But maybe we've got some Hollywood friends that understand formats of shows and what have you. What would be kind of neat, and when you were talking about the this astronomy month coming up in April, as a precursor to that, and I love when you join us, Liam, you always have some insight here and some little nuanced fact about the space station. And of course, if the ISS above starts blinking, everyone stop, because Liam's going to announce it, right? It's like it's coming overhead. But what if we did like a little segment of your night sky? So tonight, when you get out, and this is what you're going to see if you're, you know, in the Northern Hemisphere, here over North America, or hey, if you're down in the Southern Hemisphere around South Africa or around Australia, here's what your night sky looks like, and this is what we're going to look for. So go out tonight and see if you can see that.
Yeah,
right?
I'll
and
give you.
We all up there, and we see, you know, Nigel, or we see Venus, or whatever it is. And it's like, okay, we're all seeing the same thing. We're all here on this one planet, looking up at something billions of miles away, trillions of miles away, light years away, right? So I don't know, what's our night sky look like tonight, Liam?
Yeah, I don't even know whether it's clear out there tonight. It was raining like cats and dogs last night.
Oh my goodness. Well, that's good for California. We need that.
Yeah.
I'd say we probably have about 60% cloud cover. It looks like we got a little blue sky.
Yeah. Last night it was a really good visible pass though that I missed. So I always cry inside a little when when that's not possible to run outside and do that. Obviously, the other night on St. Patrick's Day, I did a little Facebook Live outside, and the neighbors next door came out in their St. Patrick's Day clothing. You know, they were literally dressed up as a leprechaun type thing, and it was really funny. And we were watching the space station as it went by, which is what you do.
Right.
Following social distancing norms, even their little 4-year-old, was following the rules and didn't run up to me and hug me like he usually does.
Oh, there you go. Yeah, that's interesting. Hey, so the night sky, if you're up for that, we could always just have a 2 minutes or 5 minutes on the night sky. Just everyone here's just a little snippet. That'd be kind of neat, or something like that. I don't know. Make it up as you will. But you and I also talked about we launched high altitude balloons, and I'm really kind of sad that we have 4 or 5 balloons that are supposed to go up between now and May, and those are all now delayed. And those large balloons have students working on the payload design, and they're contacting the Federal Aviation Administration for flight clearance of these balloons. And, it's really exciting to see the work that they do. But we also have, thanks to an engineer down at Huntsville, Bill Brown, who's been building little HABs and big HABs for 30 some odd years, something called a Pico HAB. And I don't know if you've got yours with you right there, Liam. You have your little Pico HAB around somewhere? A Sky Tracker? This thing is a super lightweight payload running on solar panels. It has a GPS on it, temperature and humidity. It has a radio, a ham radio, so you can track it. And it flies on just a Mylar party balloon. So, Liam, you and I were chatting just a couple days ago, if not yesterday. I don't know. It's all blurred to me now. Feels like I just haven't gotten any sleep. But this gorgeous little device, we launched in California, and we lost off the coast of Africa once after traveling 4 or 5 days. That little beauty right there. Working with our students, usually they do Earth investigations. And we can do these sessions with teachers like that, and they can share that with their students. And we should talk about the layers of the atmosphere, right? And that's then they learn a little bit about that. They learn about the tropopause, that intermediate layer as you go to the stratosphere. They learn about what the ozone layer does. This little guy gets around 27 to 30,000 feet, and it gets in the jet stream, and it just goes on it.
I love how it looked like a miniature space station.
Yeah. And it weighs all like 16 grams. Super lightweight. The PCB board is wafer thin. A really amazing bit of ingenuity and creativity to put that together. But just a 1 meter Mylar party balloon. You can go out and get a tank at Walmart. Well, I have some really nice helium tanks back at the office. I can probably use those. But what Liam and I were thinking, and maybe even get Bill to do one of these in Alabama, is do some sessions.
Oh, Ted, I just realized, because I think you're no one can see this. I've been lining up while you're talking. I think it's just showing you.
Oh my goodness! Oh, that's okay.
Well, if you've got gallery view, you see it all. But
there you go. Then you talk, Liam. Show it off.
Yeah. No. Anyway, the other way, there's an option you can put it back to gallery view, and then it'll show all of us. I don't know, whoever's got the host privileges on the Zoom meeting. If you switch it back to gallery or speaker view, I think it might be called. What is the one?
You're on speaker view right now. At least what I'm seeing.
No. So anyway, there's another option where it shows the the quad view or whatever.
That's the gallery view.
Gallery view, yeah. Anyway, but don't worry about it. This is what we've been talking about.
To me, it's a there's a ham radio on that, because I remember my Heathkit radio when I was a kid was like, I don't know, it was massive. Yeah. It was a good 20-30 centimeters long, and boxy, weighed about 20 pounds.
Yeah, so I've got another one around here somewhere. They normally are much larger. This is what we'd fly in like a rocket,
Mhm.
for example, or a balloon. This is another APRS radio,
Yeah, and
another version.
this is my APRS radio. Let me just switch it on.
Yeah.
So this thing communicates with this.
When
And
you got power.
in fact, I've got it on. Let me switch it to the APRS side of things. So when that's not necessarily, I haven't, let me just see.
You might need to get some juice in there through your panels first.
Yeah, that's right. I'm wondering whether it would, the
It takes a
solar
little
panels.
while. Yeah. Well, normally we'll set this up in like a
Yeah.
classroom, lower ceiling. We wouldn't do it outdoors.
Mhm.
You get neutral buoyancy first. So you start to talk about buoyancy, right? And creating positive lift or negative lift, and you get any different way you want to go there. And then we have to just have just the right positive lift. And what's cool about Bill Brown, a penny weighs 2.5 grams, and a nickel weighs 5 grams. So when he sends you your kit, he's weighed everything out, and he has a little baggie with BBs in it and pennies and nickels and whatever, just to get the right weight. So he ends up with a little extra change on every flight. But they're all just slightly different. And little 3x5 cards are handwritten out about all the specs on everything has been weighed down to the 2 decimal places in grams. And we really designed mathematically to get this just the optimal lift, so that students can study here all remotely. It's just an idea. And then we can set one adrift to LA or in Alabama or here in Berkeley, and we can watch it go for 5 days or longer as it travels across the United States. We could also even plan and look at NOAA data and a wind model and try to get the longest flight we can over the next 4 weeks, and getting everyone to prognosticate when we think we should launch and have votes and then get the thing up in the air. So clearly we're focused on our challenge here with the legumes, because we do need your help. We're going to be selecting some candidates, but we've got a little extra time now. I'm thinking, can we have a little more fun? And so I think PicoHab is calling out for us to do something cool. And I think that'd be really fun to set that thing aloft and travel. Maybe, we had one we launched from Kentucky, and it went out over the Atlantic, came down over the UK into over Spain toward North Africa. We lost it for a day. It woke up again as it was traveling north over Greece, heading toward Russia. We lost it over Bulgaria on the way to St. Petersburg. And I was like, wow, we're almost around the world. And so there are different things we can do as engineers, because he has been able to get this thing to go all the way around the Earth, circumnavigate the planet. Now, a space station takes 90 minutes, and it knows what it's doing. It's booking along at 17,500 miles an hour. Our little balloon's floating at 27,000 feet. The wind is bumping it around. The rain's coming in 3 days. It may not even make it to Utah. But it's exciting, and it could be a real fun investigation to get people involved. So I've got to check and make sure I've got that capability to pull this off. I think we do. We should be able to do that. And I'd be excited to add that to the mix of fun things we can do. And teachers out there, you got your fun thing that you like to do, you want to share, that you believe people can engage with, the teachers and students alike, that hopefully doesn't require a lot of digital. Digital's cool. And of course, let's go deep if you got it. But I'm also trying to think about those students that really are hungry for something. They can't go out and play with their friends. They're kind of stuck at home, and maybe the environment may not be the best environment. So what can we do to kind of reach out and just say, we hear you, here's some cool stuff, find a way to connect with them. I think we'll be able to connect with most if we can find cool ways to do it. So that's just another idea. Liam, you triggered my mind thinking about it. Always fun things to think about. Astronomy, this little PicoHab. Doesn't have to be related to space, robotics, I think, but how do you do robotics at home? Random sorting algorithm with a bunch of kids you can do. You can't do it by yourself though. So I guess I have to think more about if we were all students and having to stick at home for a month and we couldn't play with our friends, and our parents really didn't want us on the PlayStation or the Xbox or online, what would we could we do that was just in there in the kitchen or in the garage or in the basement that wouldn't cause too much trouble. Maybe we get a take apart day, right? Where you find something, your parents say it's okay, I'm going to throw it away anyway. And we get out the wrenches and the screwdrivers and we're just taking stuff apart. And we're trying to identify it like the way you dissected your plant.
Yeah.
We know what we're looking for on that one. But what's this gizmo? Now, there are some devices that we're going to recommend you don't mess with. Large power supplies are not pretty if you don't know what you're doing. They can discharge, and that's very dangerous. So understanding what you're looking at, an old clock, sure, why not take it apart? An old telephone, maybe you got one that still has a rotary dial. If your parents don't think that's an antique and it's okay to take it apart, ask them first. Right? But there's cool things to do. How to make a book, right?
You can learn so much by taking something apart.
Yeah. I learned that you don't need all those screws.
Because there's always a couple left over when you put it back together.
Yeah, these are extra. Where'd those come from? That's so one thing we learn is, I guess, as we start doing that, is you don't just try to remember the sequence. You write down the sequence, or any other way to remember from A to Z and from Z to A again, if you've done it in the right sequence, because it will be, what's this for? This gasket doesn't really look important, but your device may no longer work. So anyway, I've kind of talked on and on because I'm just trying to make it to 5 o'clock. 5 o'clock.
Yeah, we filled our time.
We did.
But lots of good ideas, and really, just I want to echo that, Ted. This is a chance for everybody to really tap into their creativity and their spirit of adventure, and just share your ideas and let's see what we can do.
So let's take that to heart then, Michael. Let's do I'm going to do this on Monday. I know everyone has their own things they're dealing with, even my colleagues here. So if anyone can make it, great. But I think what we're going to do is open the doors up again, digital doors, and I think we're going to figure out some of our technical issues. But it's going to be an unconference. Bring with it what you will. Keep it with lessons and learning. At home, let's try to keep consistent with the experiences we think we understand our teachers and students are going through around the world and in our country and our communities. But let's have that conversation, and no holds barred, right? Anything goes. We'll weave our leguminauts in there a little bit, but let's make you guys the star of the show. I know you've got stuff you're dying to share with us. So let's make Monday, maybe even Tuesday. Let's make the whole week. I now got October, so I've got some breathing space. So it's here, and let's get some new ideas going. I like that. We're going to continue focusing on our leguminauts. We're going to have those things filled out, some forms. We're going to finalize how we think we want to run this program, and think about if you've got some thoughts or questions or ideas around the challenge. That's probably our primary mission right here, because we want to make sure that we do have your help in getting that up. So team at Magnitude.io. So for those of you hung on through the whole experience with us, thanks for joining. Have a wonderful weekend. Make it the best. I've seen some cool little maps of like wine tours, and it's the rooms of the house, right? So if you're going to go on a wine tour, spend a little time in the kitchen, a little time in the living room, different bottle of wine. What does it pair well with? But have the best weekend you can have. If anyone has an idea or thought, I'm plugged in and thinking about what Monday looks like, and I think we're here to listen. And I know I talk a lot, but I do like to try to listen too. So if you call, I'll shut up. How's that sound? Guys, have a wonderful weekend. Michael, Scott, Liam, Tony, am I missing anybody? Did anyone else pop in? Thank you for being with us. And guys, on YouTube, on Facebook, have a great weekend. And
take care of yourselves, everybody, and each other.
See ya.
For sure. Thank you. Bye-bye.
Bye now.